Great. Good afternoon, everybody. And first, let me start off thanking Paul Allen for the foresight of funding all of this cool, interesting work. I've had a lot of fun hearing about things, and I think in particular, work that goes across fields that, at least from my vantage point, I could see using, applying, or collaborating with people who otherwise I would not be in a departmental meeting or meeting with. I also want to thank the Paul G. Allen Frontiers Group and Tom and Kathy in particular for arranging this meeting where we get to know each other as friends and family and talk about science. So I'm going to take us back to the brain and the brain of mammals, and in particular, not the brain of humans, but with goals toward that. I'm going to take you to the cerebral cortex, which in many ways, I guess, from this early or alternate logo of the Frontiers Group is sort of my Everest. So we'd like to understand, I think, in the field and in my lab, how does this amazing organ that I'm talking with you with, and that we had dinner and we told jokes and sort of wry humor and whatever we do, sits here, how does it build? How is it organized? How does it get diseased? What goes wrong? And how might you try to rebuild it in regeneration? And I think there are goals that led to our ADI cohort of modeling disease like we just heard about, modeling Rett, and modeling cortical and other neuronal diseases in culture. But I'm going to talk about applications to both understanding neuronal maturation in cultured cells and also in the brain of the genetic model mice. So my longest title ever, I try to do that at all meetings, is to talk about neuronal maturation, and I'm going to change up the order here, not of the title, but the way I say it to you, to offer an alternative or at least a complementary view to some of the things we've been hearing about so far this morning and yesterday. Rather than at least solely chronology and aging, how about if neurons have to go through checkpoints as they mature, expand, and do some very unique things? Neurons, I think, are unique except for maybe the immune system, and even then they're unique, neurons are, expand their own microenvironment as they grow and mature. They interact with new microenvironmental cells and things and places, and therefore they're never quite the same as they mature. And I'm going to propose to you a few systems to try to interrogate that, and a few bits of data that already argue that very strongly. So there are 4 parts of our overall project, and oh, is this moving, or I guess I'll never see this? I guess not. I won't know what the time is. I was counting on that. Sorry. I'm going to tell you a little bit of background, then just a couple of slides, a couple of bits of data on some already published and hopefully close to next stage published data on an engineered system to place in many, many cells, in all neurons, or I think other people in the room, in many, many or all cells in a culture or a system that could act as a molecular data acquisition and recording device, or in my view, metaphorically, like a cellular flight data recorder, to record the transcriptional or other biological history over time of each individual cell, able to be read back later on to ask who's done the right flight path, developmentally or maturationally speaking, who's done a confused one, who's done a wrong one, who's died. Timing is easy, and that already works. I won't tell you anything about that. That's just to bring chronologic time into developmental or maturational time, sort of like a race chronometer. Everybody starts when they cross the starting line, and that can be resynced with multiple onsets of different transcriptional regulators or events. I'm going to tell you about how many cells, potentially, neurons interact with during this maturational stage, and a synthetic biology tool that we're developing that's working in cells but not yet in mice. We haven't implemented it yet, that we term Hodaptics, that gets at what one could think of as the interactome of a maturing neuron, in vivo, and all of these could go in vivo or in vitro. And then I'm going to spend most of my time telling you about work the first part of which is completed, on subtype-specific machinery in growth cones. And I'll tell you the bottom line now. I'm going to show you evidence that growth cones, those extensions at the end of axons, are not just some hand waving in the breeze by the soma, but rather seem like very heavily machinery complemented autonomous minicellular units. And I'm going to show you how we got to that by higher full-depth transcriptomes and proteomes in specific types of growth cones. So many people would claim that the goal of the nervous system is motor output. I'd like to put a little more subtle touch, because I believe in the cerebral cortex, that the central function of the central nervous system, cortex in particular, is to process and integrate sensory input based on experience, hopefully with some cognition, with some memory, then into motor output. And all of these highest level motor functions rely on high level circuits of the cerebral cortex, and that's where we're most interested. Now, 130 years ago or so, the first Nobel Prize winner in the neuroscience discipline, Santiago Ramon y Cajal, our patron saint in neuroscience, described the immense diversity that underlies those kinds of complex functions, that integration of sensory domains, association, cognition. He couldn't ascribe functions to individual neurons, but it's the well-known famous diversity across layers, 6, 5, 4, 3, 2, 1, built inside out. But what most people forget usually is that he also pointed out immense diversity area to area. And there are 35 or 52 or 80 different areas in this welcome mat-sized cerebral cortex. So there are really that many different cerebral cortices, each with distinct, diverse, subtle, or grossly different diversity of neurons. And neurons are not layer neurons, because here you can see diversity of cortical output neurons to the optic tectum, here in visual cortex, I'm pretty sure, or callosal neurons to the other side. Same thing's true in motor cortex. So neurons are subtype specific. I'm not going to give you a whole cortical development or connectivity talk, but in 20 seconds, there are neurons, area specifically. So this is mouse brain, olfactory bulb, and nose that way, sagittal view, depending on area, that project out of the cortex to individual thalamic subnuclei to process sensory and other motor and other integrative behavior. There are area-specific output neurons that go to the brainstem or the spinal cord for motor or sensory function. There are commissural, or in this case, callosal projection neurons that do the point-to-point connectivity, associating, integrating across different sensory and motor and integrative modalities. There are associative neurons ipsilaterally, but the ones I want to point out to you most for this talk are underappreciated, and by many, it's a surprise. There are projection neurons in cortex in particular that have 2 or 3 or more distinct axons. And I'm not talking about a distant collateral. I'm talking about just down off cell body, 100 microns down, 2 growth cones with their filopodia sensing exactly the same microenvironmental space at the same moment in time, and one goes to California and one goes to Florida. For those of you from California, you could view it East Coast and Baja. But it works. So that raises the question of what I'm going to show you in the next slide or two. If we understand a pretty large amount, it's piddling small, real biologically, but a fairly large amount about transcriptional regulation of many of these subtypes of neurons, how does one transcriptional regulatory machinery, one nucleus, control 2 growth cones to two totally distinct things, or 3 or 4, at the same moment in time? And that suggests there might be a high level of pre-programmed autonomy. So this is the famous kind of slide. For those of you who don't recall, mouse development maybe 19-20 days, and this is mostly second half, third trimester. Diverse progenitors down near the ventricle or just outside of it generate in an inside-out fashion these many layers. I won't get into that, but we know a lot about the timing, using ways to take the cortex apart that I won't tell you about, and mouse genetics. My lab and other labs have identified a number of frequently cross-repressive transcriptional regulatory programs that direct the differentiation of newly newly post-mitotic projection neurons in what I view as an n-dimensional space. Oh, my God. Oh, this says 710. OK. I'm going to go over, but I'm going to try not too much. So those cross-repressive programs I like to put into now what I view as a modified Boolean logic of multi-stage, multi-state effect of positive and negative regulators, where the same regulators can actually be recycled and function with totally different downstream targets at a later state. So if one looks in culture, and Bill Lowry showed us some of this, and others have shown us, and does attempted directed differentiation to try to get, for example, cortical neurons, I think many people in the room and in the field would agree that current differentiation approaches from ES or IPS, whether you're trying to get neocortical-like or some other type of neuron, end up with immature neurons that have not fully distinguished themselves from other subtypes. Is that a lack of cross-repression? Is that a lack of sharpening at boundaries? Well, we'd like to put digital flight data recorders in each of those neurons as they're differentiating from pluripotent stem cells or progenitors, whatever stage, and ask, can those flight data recorders record the whole transcriptional history of that cell? It can be more than transcriptional history. I'll say the biologic history. And the requirements for that are a system that can give a linear record over time and that has enough data capacity. So in the first steps of this, a brilliant postdoc in my lab, Seth Shipman, with an LSRF, teamed up with Jeff Navala in George Church's lab. And George has been interested for a long time in molecular memory devices, and we were interested in a molecular history device, and frankly, those are the same thing. So without going through the data, Seth and Jeff were able to optimize some PAM sequences and use not the CRISPR editing side, but the adaptation side of the CRISPR-Cas1 and 2 to put in oligos and have them linearly placed into sequence to tell us a history of, in this case, arbitrary events that we told bacteria that they might have encountered. In a next step that we hope will be published soon, they've gone for higher content molecular recordings, Jeff and Seth have. And in fact, by figuring out how to code for each pixel, to do some redundancy, to code for the 4 tones or more in this grayscale, one can come up with oligos that are optimized that can encode with high redundancy whole images, read, store, expand, and then sequence, read them out, and get images with kilobytes of data. In fact, one can even do this with the famous Edward Muybridge 5 bits of the famous horse running kind of GIF. And to cut to the chase, read out of bacteria, depending on depth of 5,000 reads or 4 million reads, one gets a higher or better or more accurate resolution. So it's a fair amount of data storage that we think will be able to now be adaptable into eukaryotic cells and neurons. So I'm going to say this in about 30 seconds and move on to the last few minutes. So a synthetic biology device that we've developed in cells that I mentioned, we're terming Hodaptics, Hodos for path and haptine for touch, to investigate neuronal maturation and circuit development through the filter of an individual neuron or subtype of neurons, growing their axons over time, and over that time interacting with potentially transient synaptic partners, maybe co-fasciculating partners, maybe trophic support cells, maybe some inducers of collaterals, maybe some transient targets, none of which we have any idea about in the mammalian central nervous system. And what we've done in a nutshell simply is to come up with a multi-component hodaptic ligand system using orthogonal exotic biologies with incredibly good signal-to-noise ratio because they're not in mammalian cells, and a multi-component hodaptic receptor system with a downstream multi-component signal transduction system ending with Cre to nucleus, where we can turn all such interacting kissing cells either red or green or any color you want, or activate them or silence them, or make them null, or take a gene out, or silence a gene, or induce death. And we're interested in doing that not to just see where these guys are, but then potentially to sort them, ask who are the partners of these growing, maturing neurons. So now let me get you, I'm going to take 3 extra minutes to get to this last part and go through quite quickly, because this still says I have 3 minutes, so I'm going to just go with my misperception view of life. There's this long-standing problem in the nervous system of the sorting problem. How do all these axons at the same time sort across one another to set up their perfect circuitry? And there's remarkable somatotopy. This is an axial section of the forebrain, where if we just put dye or if we did this genetically, there's a perfect alignment of neurons going to their point-to-point connectivity. So some people have thought, well, maybe this is like a computer cable, and first one in is up at the top of this trajectory, and then the next guys just fasciculate on. But it's really too perfect for that, and I'm going to show you direct evidence that that doesn't seem to be the case. And Alex Palopoulos and Alex Murphy, brilliant current postdoc and recently defended graduate student, took on this project and wanted to see growth cones, but not in culture as they've been studied with about half a dozen locally translated proteins identified, but directly out of the brain. And we know that growth cones extend from these projection neurons for days and days and days, 10 to the 3, 10 to the 4, 10 to the 5 cell body diameters away, where retrograde axonal transport would take 8 to 15 hours to even signal to the nucleus. So first, to see whether some of that information that I told you about with the somatotopy is intrinsic, we did a simple old-fashioned experiment and took some red and green mice and took lateral and medial callosals that should go to mirror image targets, shook them up in a jar, plated them down, they're all healthy, and then heterotopically transplanted them into medial cortex. And if fasciculation is the key, these guys should all go to medial cortex, but they don't. The green guys that started off here go to their appropriate lateral targets. The medial guys go to the right targets. So that says the neurons know who they are. So we came up with ways of isolating growth cones from genetically or molecularly subtype labeled cells. This is in a whole brain, so everybody else in the whole brain is dark. We can take these growth cones out. This can be from physical or genetic Cree lines or electroporation. Because of time, and I'm over, we can use an old 1983 from when I was a grad student EM isolation protocol for growth cones. I colored them, so they're not colored in the EM. We can do a fractionation, a biochemical isolation. We then teamed up with BD to come up with modifications of FACS sorting, but for small particles, and can purify those red or green or whatever colored growth cones out of the black background. They're all growth cones. I can tell you molecularly they're enriched. They have high-quality RNA and protein for fuller high-depth proteomics and transcriptomics. They're 300 to 800 nanometers. They're the right size. They hold up to trypsin and RNase protection, so we know we have closed spheroids. We can get rid of everything on the outside. We're still in the linear range of getting more counts, but we get saturated with hits, so we think we're pretty deep in our proteomics and transcriptomics. And let me just tell you in 30 seconds 2 bits of biology. We can isolate the growth cones, as I've told you, and their parent cell bodies simultaneously, and do what we are thinking of as subcellular RNA-Proteome mapping, where in this vast expanse of a cell, 10 to the 4 cell body diameters away, with 95 or 99 or 90% of the cytoplasm in the axon, not in the cell body, where is the actual machinery for certain circuit decisions happening? And what we find in growth cones, for example, here's at the proteomic level, is that they have full minicellular kind of machinery. They have full ribosomes, but not normal somatic ribosomes. They have proteasomal and lysosomal machinery. Of course, they have all the receptor cytoskeleton stuff we'd all expect, and actin, tubulin, chaperonin kind of pathways. They have energy, but they also have a lot of RNA binding and some very specific things that one doesn't even see in the cell body by orders of magnitude. And at the transcript level, the same thing is true. There are hundreds of transcripts enriched hundreds, orders of magnitude over cell bodies, so you wouldn't even see them by deep sequencing. So I view these as essentially NASA-programmed deep space satellites, one going into the sun or Mercury, one going out to the solar system. They're going to have some common machinery, maybe propulsion, maybe solar panels, but different sensors and different autonomous kinds of programs to follow. But then they're going to have to make their decisions on their own. And in fact, that's what we do. We've looked at 3 distinct subtypes now. And if we do that, even between the very closely related medial and lateral callosals, there's core machinery and then distinct biological differences that we think underlie some of this somatotopic organization. So after early programming by the nucleus, growth cones appear to potentially be quite autonomous minicellular units that might directly control subtype-specific development, maturation, interaction with that interactome, I think regeneration, and maybe underlie aspects of degeneration by early vulnerability and the fact that growth cones turn into synapses, and those are what frequently falls apart in degeneration. And the striking thing is the molecular networks I'm telling you about a little bit are not even observed as somal transcripts. So if we do full transcriptomes of cells in culture from somas, we're not even going to see maybe much of their operational circuit building, maintaining, and functioning machinery. I think this is applicable to the kinds of dysmorphic, non-regenerating growth cones that Santiago Ramon y Cajal described 100 years ago or more, and also to the polygenic human genomics of intellectual disabilities and autism spectrum disorders, where so many of the hits are coming up in what we all think of as synaptic proteins, but could those also be developmental growth cone turning into synapses and setting those up kinds of proteins? So I think I'll end there. And former members of the lab, I only showed you work of the 3 yellow bolded people. I've shown you the colleagues and collaborators. And I want to thank again the Paul G. Allen Frontiers Group for supporting this kind of non-hypothesis-driven discovery work. So thanks very much, and I'm sorry to have gone over.
We have time maybe for one quick question, if there is one.
Are the surface proteins different in the growth cones?
Yes. So some of that non-core machinery that I was showing you on that volcano plot without getting into details, the sort of low-hanging fruit are surface molecules. And what one might think the obvious things that those might help with self-organization and things like that.